Hello, everybody. And as we continue our journey through genetics, we're going to talk about linked genes. So up until now, all of Mendel's work basically assumed that the genes that he was looking at were on different pairs of homologous chromosomes. Okay, so in order for independent assortment to work and the law of segregation, those genes needed to be and were on different chromosomes. Now, this is not always true. So sometimes genes are on the same chromosome. Actually, a lot of the time, because there are thousands of genes on every chromosome. Okay? So if you have alleles that are on the same chromosome, they do not exhibit independent assortment. They're not separated at anaphase 1. So they travel together. It almost is like they are linked up. And so we call this phenomenon gene linkage. So if you have genes on a chromosome that tend to be inherited together, it's called linked genes or gene linkage. Okay? So after Mendel came up with all these laws, and again, he was pretty lucky because everything, behaved according to Mendelian genetics because he was always looking at genes that were not linked. But it turns out genes are linked. So quick example to show you how this looks is that if you have the gene for flower color and pollen shape, okay, and this is, first of all, it's saying they're linked. Their loci are on the same chromosome. Okay, so we have purple dominant to red, long dominant to round for pollen shape. Here's our alleles. Capital P is purple, lowercase p is red. Capital L is long, lowercase l is short. So if we have this parent here, okay, we would predict, knowing what we know about dihybrid crosses, that the independent assortment would yield the following gametes, okay? So 2 dominant alleles, 1 dominant, 1 recessive, the other dominant, the other recessive, and 2 recessive. And they'd be about even distribution. You get 1 out of 4 of these, 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 etc. However, with these 2 genes, this result here is not what they observed, okay? Instead, the gametes were pretty much all 2 dominant alleles together and 2 recessive alleles together. So it was hypothesized that these guys were traveling kind of as a package deal, and they were linked. And so you don't get kind of the expected results you would from Mendelian genetics. They don't behave the same way. Okay. So again, you would have these 2 genes, alleles on the same chromosome, and they would stay together because they're on the same chromosome. Same thing with the 2 recessive alleles. They're on the same chromosome and they stay together. They travel as a pair. Okay. Now, do you think there's any way that these alleles could be separated from each other? What do you think? Oh, that's right. Recombination and crossing over. So if these homologs were to recombine, you could switch these 2 portions of chromosomes, okay, and then get new combinations of alleles. Okay, so we're going to look a little bit at that and then maybe come back to this. Okay, so crossing over creates new combinations of linked alleles. Okay, genes on the same chromosome, do not always stay together. Sometimes crossing over occurs between pairs of homologous chromosomes. Okay, and again, during synapsis. And the interesting thing about this is that basically the farther apart 2 gene loci are, the more likely crossing over is to occur between them. So this means if I have a chromosome here, woo, and let's say I have gene A, B, and C. Between gene A and C, there's more distance, so there's more opportunities to cross over. However, between genes B and C, they're much closer together, So there's fewer opportunities. Okay. So B and C would be more tightly linked than A and C would. Okay. If the loci are very far apart, crossing over occurs so often between them that they actually behave as if they are on independent chromosomes, so that they behave as if they're on different chromosomes. Okay. But if the genes are closer together, they travel together. Okay. So again, crossing over, recombination generates new gene combinations. Okay. I want to talk just for a second about this here. So A, B, and C. So again, we can look at the frequency of recombination between A and C. There's more space, so it would occur more often, let's say 45% of the time. Okay? Between B and C, they're closer together. So B and C are separated only about 10% of the time. Okay? And this is called the recombination frequency. And because recombination frequency corresponds to how far apart genes are on the chromosome, they can be used to measure distance. Okay, and we call this distance. Okay, so 1% recombination frequency is equal to 1 map unit, which is a unit of distance on a chromosome. And the map units are measured in centimorgans. Okay, C, oops, that C was too big. Small c, capital M, centimorgans. Okay, and we'll go over how to use that information to do gene maps in class.